Assalamu alaikum. How are you doing, dear students? This lesson will be our final lesson in Chapter 2, titled by Activity 4, Identical Reproduction and the Cell Cycle. At the end of this video, you will be able to recognize that the cell cycle consists of interphase and mitosis, infer that DNA replication occurs during S phase of the cell cycle, describe the steps of DNA replication, deduce that the DNA replication is semi-conservative, and explain the variation of the DNA amount per cell during the cell cycle. Don't forget to prepare your notebook in order to take notes. This cell is found during mitosis, having chromosomes with 2 chromatids. However, after and before mitosis, this cell normally have chromosomes with only 1 chromatid. So how is this possible? How does 1 chromatid become 2 for each chromosome? The solution lies in the process happening before mitosis, the interphase. So our talk in this lesson will be mainly about what's happening during interphase. When checking again the title of this lesson, we find the important term, the cell cycle. Let's see what's a cell cycle. Looking at this cell cycle here, we find mitosis that results in 2 daughter cells after division. And of course, interphase that occurs before mitosis. As we divided mitosis into prophase, metaphase, anaphase, and telophase, we will also divide interphase into G1 or gap 1, S phase also known as synthesis, and G2 also known as gap 2 phase. And this is in short the cell cycle. Now let's concentrate on interphase, where this cell prepares itself for division. How and what's happening in each step of interphase? To find the importance of each step, I must ask for your help. Pause the video and compare each cell before and after each step, gap 1, synthesis, and gap 2. What do you notice? After comparing the steps, you can find that during gap 1, every organelle is duplicated. For example, the vacuole here becomes 2, the mitochondria here becomes 2 also, and every organelle is being duplicated. So in gap 1, the cell copies organelles. And what's happening during synthesis is that the amount of DNA found in the nucleus here is being doubled. So here, the DNA duplicates. And finally, in gap 2, nothing important is happening inside the cell. However, what's happening is that this cell enlarges so that the cell will grow and certain enzymes are prepared to help in division. For now, what's happening during gap 1 and gap 2 is clear. During gap 1, the cell copies organelles, and during gap 2, the cell enlarges. However, during synthesis, how is this DNA inside the nucleus is being doubled? What's exactly happening here is in our next title, DNA replication. Please watch the following video and find the purpose and result of this DNA replication occurring during synthesis.
During DNA replication, the double helix unwinds. Then, an enzyme called DNA helicase unzips the DNA, so the 2 strands are separated. This happens at several points along the DNA. An enzyme called DNA polymerase attaches itself to the DNA strands and is used to add complementary free nucleotides to the now exposed bases on both strands. A pairs with T and C pairs with G. This forms 2 DNA molecules, each of which have 1 brand new strand and 1 from the original DNA. These 2 strands twist to form a double helix. The 2 new DNA molecules are both identical to the original DNA molecule.
It's clear from what you've watched in the video that the purpose of this DNA replication is to copy the strands of DNA, where in the result you will get 2 identical molecules of DNA as you're seeing in the adjacent figure. So DNA replication is the biological process to produce 2 identical molecules of DNA starting from 1 original DNA molecule. Those 2 strands of the DNA molecule will become 2 brand new DNA molecules, each one identical to the other. So let's list the steps of DNA replication, where in the first step, the 2 strands of the double helix unwind. An enzyme called DNA polymerase binds to each strand of the DNA molecule. Then this DNA polymerase will start replicating or duplicating the strands of DNA by adding complementary base pairs. A is complementary to T and G is complementary to C. Then finally, you will get 2 identical molecules of DNA. Now, I will ask for your help again. Please compare the old strand and the new strand of the DNA after replication. There are two important things to notice here. What are they? Let's first start by comparing the two strands of the same DNA molecule. And as we've been saying from the beginning of the video, that each strand is complementary to the other. T is complementary to A in the second strand and C is complementary to G in the opposite strand. And now when comparing the two DNA molecules, you can find that they have the same sequence, where the new strand in one DNA molecule is the same as the old strand in the other DNA molecule. The sequence TG is same as the sequence in the other strand TG. And for now, you understood the parts of the cell cycle. You defined the DNA replication and described each step. Now let's deduce the form of DNA replication. Scientists proposed two models of DNA replication. The semi-conservative, where the resulting daughter DNA molecules will have one new strand and one old one, and the conservative model, where you have one totally old DNA molecule and one totally new DNA molecule. The old one is conserved. By looking again at this image, what is the correct model of DNA replication? Is it semi-conservative or conservative? Remember, the two resulting DNA molecules have one old original strand and one new strand. Well, it's obvious that the correct model here is semi-conservative, where one strand of DNA is conserved and the other strand is the new replicated one. So in short, you have this cell, and before S phase of interphase, it contains simple decondensed chromosomes, each with one chromatid, and one chromatid refers to one DNA molecule. During S phase, you will get replication fork. A replication fork is formed when the DNA unwinds and replication is occurring. And finally, after S phase or during G2, you will get double decondensed chromosome, each with two chromatids. Two chromatids refer to two DNA molecules. Here we've answered our important question: how do we get two chromatids for each chromosome? The answer is in DNA replication, where you had chromosomes with one chromatid or one DNA molecule. After DNA replication, this chromosome will contain two chromatids or two DNA molecules, and finally, after division or mitosis, it will return to its original state, having one chromatid or one DNA molecule. So how does this amount of DNA vary during the cell cycle? First, you had this cell. This cell originally or during G1 will contain a chromosome with one chromatid, which refers to one DNA molecule. However, when this cell passes through the S phase, replication fork will occur where DNA is being replicated. As a result, and after S phase, this cell will contain decondensed chromosomes with two chromatids, which refers to two DNA molecules. Now finally, when passing through mitosis, the cell will give two daughter cells, each having one copy of the DNA molecule, which refers to one chromatid per one chromosome. Now let's explain the idea again graphically, where you have this graph that represents the amount of DNA in a cell progressing through the stages of the cell cycle. Pause the video and remember what we've explained, then try to label the line segments between those hatch marks to show G1, S, G2, and mitosis. Where does each step or phase occur? As we explained previously, the first step of the cell cycle is known as G1, where the quantity of DNA remains the same per cell, Q. This quantity increases when DNA replication occurs during S phase. After S phase, the amount of DNA per cell will become 2Q, double the amount of the original DNA quantity. And here comes G2. After G2, we have mitosis, where after cell division, everything will return to its original quantity, Q. So here we've listed simply the steps of the cell cycle, referring to the variation of the quantity of DNA in progress with time, G1, S, G2, and finally mitosis. Remember that during G1 we have decondensed chromosome with one DNA molecule, one chromatid. After S phase and during G2, you will get 2 chromatids or 2 DNA molecules per chromosome. And finally, after mitosis, this quantity will return to being 1 chromatid per 1 chromosome. So you can conclude that the starting amount is the same as the final amount. After mitosis, the original quantity of DNA is maintained. And here we've summarized the steps of the cell cycle graphically. In this video, you've learned that the cell cycle is of 2 parts: interphase G1S G2 and mitosis with its several phases. During S phase, DNA replication occurs where DNA is duplicated. The phases of DNA replication are summarized by: first, the double helix unwinds, then DNA polymerase binds to both strands of the DNA. DNA polymerase replicates the DNA by adding complementary bases, and finally you will get 2 DNA molecules according to the semi-conservative model. As an assignment, please make sure to memorize the summary sheet. This video is prepared and recorded by teacher Zahra Ashari and supervised by teacher Ihsan Hassan. Thank you for your time.